Hi guys, it's Mr. Y. Today we're going to be talking about cellular respiration. Sorry I didn't put a title on this. So, respiration. And before we begin, I wanted to let you know that these links here, which obviously you cannot click on, I will put as many of them as I can down in the description of this video. So either before or after you're done watching this video, I want you to click these links, these here in the blue. So just check the description below and you should find them hopefully working, and watch them all. They all have different animations for concepts I'm going to be talking about. So if at any point you want to stop the video and go to a concept like glycolysis, citric acid cycle, electron transport, or ATP synthase, and click on the appropriate link to see what I'm talking about more fluidly, by all means do that. Okay, to begin with, we actually have to start with a new molecule that's going to show up a lot in cell respiration and again in photosynthesis in a variation. That's called NAD+. Now, NAD+ I've drawn here as this little guy with these little open spots here, these little active sites. And right now they are empty. And you kind of see it kind of looks like a car that's upside down without wheels. But keep in mind, NAD+ actually has a very important function. Its function is to carry things around, specifically pairs of electrons. So those are two electrons. Now, when it's full, NAD+ becomes NADH. The H stands for the proton ions. So NAD+ and NADH are just the same thing. It's just one version. The NAD+ is empty. The NADH is full. So you can always tell whether it's a full molecule or an empty molecule by the name people refer to it as. They always are transporting electrons, and they do have another variety called FAD and FADH2, and they work under the same principle. They're carrying electrons, and when they're called FAD, they're empty. When they're called FADH2, they are full of pairs of electrons, and we'll see those again when we talk about photosynthesis. And one other very quick lesson, that's how do we actually get electrons to be by themselves? And from our chemistry unit, you should have learned that hydrogen, being a very simple atom, is composed of just one proton and one electron. So one easy way to get a proton by itself is just to take away the electron, and then you just have H+. That's a hydrogen atom, hydrogen ion, excuse me. And then you are also left with an H-, which is basically an electron, or an E-. So usually we'll use this symbol there for electrons. So a lot of times in diagrams you'll see this H+ referred to for protons, and then you'll see the electrons show up as well. Okay, now cell respiration. This is how eukaryotic cells, cells with nucleuses, actually make ATP. Remember, ATP is energy for the cells. It is the cash that they use to do all the internal work that they have to get done and to actually be able to make certain enzymes work as well. It's how basically plants and animals take the ingested food, and not plants obviously, they do photosynthesis, but they take the energy from the sunlight and actually turn it into workable versions to be used throughout the cell. Now, this is just the general formula, which you should have learned in middle school. Oxygen plus food gives you carbon dioxide, water, and energy. Now, down below, we have it slightly more specific again. Oxygen plus food. In this case, I put glucose, which is just a sugar. That's glucose. Gives you carbon dioxide, CO2, water, H2O, and ATP. And then down below, this is going to be the version that I'm going to want to make sure you guys learn, because this is actually fairly balanced. Not entirely, though. We'll see why in a bit. 6 oxygen plus a glucose molecule gives you 6 carbon dioxide, 6 water molecules, and about 36 to 38 ATP yield. That's how much energy you produce for every 1 glucose. Okay, so this picture I like because it shows all 3 stages of cell respiration and where they are happening in each stage. So I would strongly recommend pausing the video at this time and copying down everything on this picture. Just even if your drawing skills are not good for the background stuff, get everything. Even get the little mitochondria here in the back so you're clear. Get all the arrows, just get everything. It will help you, trust me. So we're going to start with stage 1 now. Stage 1 is glycolysis. You can see that happens inside the cytoplasm in the cell, inside the fluid medium of the cell. So this is not inside of the mitochondria yet. We'll eventually get there later. And you can see we're going to start with glucose, sugar, and it will end up with this substance called pyruvic acid. Pyruvic acid. So stage 1, glycolysis. We start with a 6-carbon chain of glucose, so 6 carbons right there, C6H12O6, and you have 2 empty electron carriers. Now, that's what goes into glycolysis. What comes out is 2 pyruvate molecules, and you'll notice each pyruvate is only 3 carbons each, and you get 2 full electron carriers, now NADH, and you also get a net gain of 2 ATP for this process. Now, the pyruvates are not in their final form. They're going to be slightly modified and turned into this stuff called acetyl-CoA, which looks like this. And there's going to be 2 of those as well before they go on to stage 2. So 2 things go in, 3 things go out. 3 things that come out, you have your ATP, 1, your full electron carriers, 2, and then your acetyl-CoA or your 2 pyruvates, depending on your perspective, 3. And then again, glucose and NAD plus are what go in. 2 NAD pluses are what go in. So what I would recommend at this point is to just pause the video and go and watch the animation link labeled glycolysis to see how this process works in motion. Again, we're going to be getting 2 ATPs out of this whole process, and the acetyl-CoA we produced is actually going to be what enters in to stage 2 called the citric acid cycle. But for now, we're done with glycolysis. Now, one word of clarification: the citric acid cycle is our 2nd stage, but your book will call it something else. Your book will call it by its alternative name, the Krebs Cycle, named after the scientist who helped discover it. So Krebs Cycle, citric acid cycle, it is the exact same thing. Please don't let that confuse you. Notice that the citric acid cycle is the first stage of cell respiration that actually happens inside the mitochondria once again. So whereas glycolysis were happening in the cytosol or cytoplasm, the citric acid cycle or the Krebs cycle is the first stage to happen inside the mitochondria. All right, so we get the acetyl-CoA from outside the mitochondria into the mitochondria. We're also going to be needing a bunch of empty electron carriers, so we have 6 NAD pluses and 2 FADs. Again, these are just the empty electron carriers. The cycle is going to turn. Because it is a cycle, it kind of repeats on itself. And for each turn of the cycle, we use up 1 acetyl-CoA. So the cycle must turn actually twice, 1 for each of these 2 acetyl-CoA. And the 6 and the 2 is actually an overall figure, so that's for overall. You usually use 3 and 1 for 1 turn, but really it's 6 and 2 for the NAD plus and the FAD. What's going to come out of the cycle when we're all done with these 2 turns is 2 ATP as a net gain, 6 NADH and FADH2. Again, my electron carriers are once again filled up. You get a waste product you guys should recognize, CO2. This is actually where the CO2 comes from that you exhale when you breathe in through your lungs. It comes down through your cells after they do the citric acid cycle, and they get rid of it, and then it gets shipped to your lungs via red blood cells, and then you exhale that. And you also get out a 4-carbon chain, and there's actually 2 of these guys. They're called oxaloacetate, and there's 2 of these guys. Again, this is what the citric acid cycle actually looks like. There's a version of this in your book on page 227. Again, they will call it the Krebs cycle. It is the same thing as citric acid cycle. Don't let that confuse you. I recommend just watching the animation that's linked below in the description. It makes it much more fluid. You do not have to worry about memorizing all this stuff, but I do want to emphasize a few key points from this picture. First, you can see here's where I have my electron carriers being filled up. The NAD pluses becoming NADH, and the FAD over here becoming FADH2. So this is where my electron carriers are getting filled. You can also see I am changing an ADP to ATP, so that's where my energy is starting to come out. And there's the oxyl acetate at the end to be recycled to the next turn of the citric acid cycle, or the Krebs cycle. So we're going to move on to the third stage because we've finished glycolysis, and you should have watched the animation on the citric acid cycle. They've both produced some ATP, not a whole lot though. So now we're going to go on to the main stage, the third stage of cell respiration. This has two parts, one part called electron transport and the other called chemoosmosis. The whole thing together, these two together, are called oxidative phosphorylation. It's a big, ugly sounding name. Don't let it scare you guys. It's really not all that complicated. You'll notice at the top of this diagram, this is one of the other reasons I wanted you to copy this diagram, it has these arrows showing that in the third stage is where we are shipping all these full electron carriers we've been stacking up. So they get shipped from stage 1 and from stage 2, the glycolysis and the citric acid cycle, to this third stage to be used to help make the ATP. That's why the ATP sign here is much, much bigger. So the first tricky part about the third stage is that the last chemical, carbon chemical we ended with, oxyl acetate, is not really important. It's still back in the citric acid cycle, repeating itself. Remember, the cycle keeps going in a circle. So oxyl acetate is not going to show up here. What is important is, as I said before, the electron carriers, all the FADH2s and the NADHs that have been filled up. These are what actually trigger the third and final stage of cell respiration. So be aware, oxyl acetate, the carbons, don't try to follow the carbons past stage 2. It won't help you. You have to follow the electron carriers. Stage 3. All right. Oxidative phosphorylation. Again, we have a whole bunch of electron carriers that have been shipped in from glycolysis and the citric acid cycle, and we're going to use those in conjunction with some oxygen, and this is the oxygen you would inhale when you breathe. We're going to run it through an electron transport chain that I'm going to show you a picture on in the next slide, and this electron transport chain exists in the mitochondrial membrane because you need to know where each of these stages is happening. And when it comes out, what you're going to see is the electrons are all gone. They've been taken off their carriers. So these carriers are now empty. They can be recycled back to glycolysis and the citric acid cycle. We're also going to get out some water here. So some of the oxygen has actually been changed into water. And we're going to get out a whopping 34 ATP. So this is where the vast majority of ATP comes from in cell respiration. I like this picture. Don't worry about reading it unless you really want to, but I like this picture because it's a beautiful drawing and it shows where the electron transport chain actually exists in the cell along the mitochondria. So it's in the mitochondria and it's in the inner membrane, because remember, the mitochondria has an outer membrane here and then it has an inner membrane here. So the electron transport chain always takes place along the inner membrane. Now we're going to go through these steps, but again, if you start getting confused, watch the video description in the description down below. It should help. Okay, I like this picture too because again, it emphasizes that we're right here on the inner membrane of the mitochondria. Now, there's several different proteins in the inner membrane of the mitochondria, and their main job is to do two things. First, they're going to take the electrons off of our electron carriers. And as they pass the electrons on along these lines, they use the energy from the electrons to push protons, remember H pluses are just protons, across the membrane. And they just take them from one side, so there should be a little H plus here, and they just scoot them on over to the other side. And then they keep passing the electron on again and again you pass a proton over, and again they pass the electron on, and again you push a proton over. You notice this last stage, oxygen shows up and you get water. And then this final stage we'll talk about on the next slide is where the ATP actually ends up being produced. So here's yet another picture of the electron transport chain. It's good. I like the other pictures a little bit better, but this is the picture you would find on page 228 of your textbook. So if you wanted to follow along with that, it's page 228. And once again, notice that here's our electron carriers, and they're emptying their electrons to these proteins stuck in the inner mitochondrial membrane. And as the electrons are moving along this path in the white here, they're giving just enough energy to these proteins to have them pump protons across the membrane. So every time one of them touches, more protons are being pumped across the membrane. And so after this has happened for a while, we are getting a bunch of protons inside and very few protons out here. There's none. And this creates what we call a concentration gradient. That means there's a very strong amount of pressure for the protons to try and equalize themselves out, but they can't cross the cell membrane. Remember, the cell membrane often prevents things with charges from going across at will. They can't do it. They're being blocked. But we'll talk about it in the next slide. The protons are being built up on the outside here as the electrons are being passed on, and you'll notice that the last the last protein in the membrane to gain an electron actually takes that electron and combines it with a few more protons that are left over, if it can find any, and oxygen. And then what comes out is water. That's where the water comes from. You combine the protons with oxygen and electron to equalize themselves out, and you get some water molecules. Now again, the protons are still trying to get inside. And as they try to get inside, the only way they can get inside is through this protein over here. This is a very special protein called ATP synthase. Now there are animations in the description down below, and I highly recommend, again, watch the one. It should be labeled ATP synthase. And this thing works very much like, if you've ever seen one, an old-fashioned water wheel. It just works sideways. Or you could even think of it like a pinwheel. As the protons are coming back through, remember, they're coming back with a lot of force now because they want to try and equalize themselves out, but this is the only way they can get back through to the other side. This part here actually rotates. It begins to spin again and again and again. And as it does that, it actually forces together an ADP and a phosphate ion to make ATP. Again, it's kind of like a water wheel in a weird sense. And so that's where a good portion of the ATP is coming from. Here is another picture showing you ATP synthase. You can see the little white dots here represents the protons trying to come across. You can see it's not a direct path, but there is a pathway for protons to come across. And this whole system rotates down here, forcing ADP with a phosphate to create ATP. That's why it's called ATP synthase. Synthase means to make ATP. So that concentration gradient, the proton concentration gradient, is what powers this thing. So we're building up the protons on this side with the electron transport chain to force this thing to work. Now, sorry, I had to wait for the bell. The key points, again, the electron transport chain is in the inner mitochondrial membrane. It's what creates the concentration gradient on the protons, for the protons outside of the inner membrane, so that the ATP synthase can work. The last part of the electron transport chain, the last spot the electron goes to, you need to have oxygen. If there is no oxygen, the electron transport chain will plug up. It will stop. And if no electrons are moving, no protons get pumped across. And when you do have oxygen, the oxygen combines with some protons and some of the electron from the electron transport chains, and you get water. Again, no oxygen, the chain stops. And the protons themselves, the H pluses, can only recross through ATP synthase, and ATP synthase, like I said, works kind of like a more complicated water wheel, but it's the same basic principle. And as it turns, it's forcing together ADP and a phosphate ion to create our ATP, our energy. So once again, guys, I recommend going back, watching any of these parts again that you need to, and then watching in conjunction any of these links in the description. They should be labeled hopefully the same thing as they're labeled here, just so that it makes sense to you. It's much better to see this in motion. Some of the links will have like steps. It'll say like step 1, 2, or 3, and you have to click the steps to make it move. So just be aware of that. So click all around, experiment, and let me know if you have any questions in class.